Hello, we're glad you've joined us for this live webinar, Highly Multiplexed Single-Cell Westerns for Exploring Heterogeneity in Protein Kinase C Signaling and CRISPR Knockdown Efficiency. I am Judy O'Rourke of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. It's brought to you by Protein Simple. Protein Simple is part of the Analytical Solutions division of Bio-Techne. Protein Simple is rethinking protein tools and helping thousands of researchers around the world resolve their protein analysis problems so they can reveal new insight into proteins and their role in disease. The company's wide-ranging portfolio of tools includes everything from immunoassay systems that quantify protein expression to systems that probe the structure and purity of protein-based therapeutics. To learn more, visit www.proteinsimple.com. Let's get started. You can pose questions to the speakers during the presentation while they're fresh in your mind. To do so, simply type them into the drop-down box located on the far left of your screen labeled Ask a Question and click on the Send button. Questions will be answered after the presentation. To enlarge the slide window, click on the arrows at the top right-hand corner of the presentation window. If you experience technical problems seeing or hearing the presentation, just click on the Support tab found at the top right of the presentation window, or report your problem by typing it into the Answer a Question box located on the far left of your screen. This is an educational webinar and thus offers free continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process of obtaining your credits. I now present today's speakers, Noemi Kadei, MD, Staff Scientist for the Collaborative Protein Technology Resource in the Office of Science and Technology Resource at the Center for Cancer Research at the National Institutes of Health, and Kelly Gardner, PhD, the Director of Marketing for Milo at Protein Simple. Dr. Kadei received her MD from the University of Debrecen in Hungary and did research for several years at the Department of Biochemistry, focusing on the role of tissue transglutaminase in apoptosis. She joined the Laboratory of Molecular Mechanisms of Tumor Promotion at the National Cancer Institute in 1999, first as a postdoctoral fellow and later as staff scientist. About a year ago, Dr. Kadei joined the Collaborative Protein Technology Resource, where she leads the efforts of the laboratory to implement novel technologies for protein detection at single-cell level, including single-cell Western and multiplex immunostaining using CODEX. Dr. Gardner was formerly the CEO and co-founder of Zephyrus Biosciences, the life science startup company that originally developed the single-cell Western product line. For her work leading the company from incorporation through to its acquisition by Protein Simple, she was named one of the top 35 entrepreneurs under 35 globally by the MIT Tech Review. Dr. Gardner holds a PhD in bioengineering, an MBA equivalent, and a Bachelor's of Science in Biomedical Engineering and Mechanical Engineering. The speakers' complete bios are found on the LabRoots website. Dr. Kadei and Dr. Gardner will now begin their presentations.
Great, thank you so much. Welcome to today's webinar presentation. My name is Kelly Gardner, Director of Marketing for the Milo Single-Cell Western platform at Protein Simple. And I'm really pleased to introduce Dr. Noemi Kadei from the Center for Cancer Research at the National Cancer Institute at the NIH, who will share some of her work developing highly multiplexed single-cell Westerns for exploring heterogeneity in protein kinase C signaling and CRISPR knockdown efficiency. Our learning objectives today are first to learn how single-cell Westerns work and how single-cell Westerns help you uncover new insights into your samples. Then Dr. Kadei will present data from 2 active projects at the National Cancer Institute. In her presentation, you will learn how her group is using highly multiplexed single-cell Westerns to study heterogeneity in signaling pathways to identify cell subpopulations with distinct activation states. You'll also learn how her group is using single-cell Westerns to measure CRISPR-Cas9 knockdown efficiency to facilitate clonal selection of knockdown cells. So if you're not familiar with single-cell Westerns, we wanted to start with a brief introduction to the technology. Milo is the world's only single-cell Western system. It brings the power and ubiquity of Western blotting to single-cell analysis. It provides Western-based protein measurements plus heterogeneity information about your sample. To give you an idea of what the platform can do, Milo can analyze about 1,000 single cells per chip. It can multiplex to measure multiple proteins per single cell. Typically, people are doing about 4 proteins per cell per probing cycle, but Milo can do 12 plus proteins per cell when including stripping and reprobing the chips. Milo offers time savings compared to conventional Western blotting. The total workflow is about 4 to 6 hours, with 3 to 4 hours of that time being hands-free. And finally, Milo uses conventional Western antibodies, so you don't need specialized reagents to measure your protein targets of interest. In fact, being able to use the huge commercial catalog of Western antibodies makes it possible to really expand the extent of the proteome that can be measured at the single-cell level. And it provides the flexibility required to measure diverse proteins at the single-cell level, including post-translational modifications like phosphorylated proteins, transcription factors, and even methylated histones involved in signaling pathways, as Dr. Cadet will discuss in her work. The single-cell Western system itself is composed of the Milo instrument, which is a benchtop instrument where the single-cell Western assay is run, a fluorescence microarray scanner, which is used to image the chips, SC-West consumable kits, which contain SC-West chips and all the buffers you need to run your assay. The full single-cell Western assay is run within an SC-West chip, which is based on cutting-edge microfluidic technology. Scout software automates the image processing and data analysis. And as I mentioned, Milo is an open platform, and so off-the-shelf antibodies can be used for probing. Bio-Techne is one of the world's largest antibodies providers, and we provide high-quality antibodies from 2 of our sister brands, R&D Systems and Novus. You can also continue to use your favorite antibodies you're already using in your lab. And our organization also offers world-class support to all of our customers. So how does the single-cell Western assay work? The SC-West chip is a 1 by 3 inch glass microscope slide with a very thin polyacrylamide gel layer on the surface and 6,400 microwells patterned into that gel. You can kind of see them in the picture there. They look like lines, but they're actually individual microwells. So we take a cell suspension and pipette it on top of the SC-West chip. And Milo can run any non-fixed sample that you can get into single-cell suspension. So this includes suspension cells, of course, but also trypsinase adherent cells or dissociated tissues. From there, the cells are settled by gravity into the microwells. And we use a Poisson distribution to capture about 1,000 single cells in a chip, and use cell concentration and the settling time to tune the chip occupancy. Then we lyse the cells in the array, and we do an SDS page separation on each single cell lysate. We then use UV light to activate a chemistry incorporated into the polyacrylamide gel, which immobilizes the proteins into the gel. And this really eliminates the traditional transfer step of traditional Westerns. Then you can probe with conventional Western antibodies by diffusing them into the gel. We typically use an unlabeled primary antibody and a fluorescent secondary, and then image the chip fluorescence. Milo runs 1,000 single-cell Westerns in parallel. So here you can see about 1/16 of an SC-West chip on the left, which contains 400 microwells. And you can see here an array of single-cell separations. If you look at the zoomed-in image on the top right, you can see 6 electrophoretic lanes, where the microwells are located at the top of each lane, and then electrophoresis is happening downward. In this view, there were single cells captured and analyzed in 4 of the 6 lanes. So each of these 4 lanes contains a single cell lysate, and this particular chip has been probed for 3 different protein targets using 2 different spectral channels. The fluorescence plot at the bottom is generated by Scout, the image analysis software, which identifies the 3 protein peaks at varying migration distances, which are proportional to each protein's molecular weight. Scout software can quantify area under the curve for each detected protein peak, which gives you quantitative information about protein abundance for each probed target in each single cell. Scout's a very powerful tool, and it really helps you to automate your image analysis. So how it works is it finds all 6,400 lanes on each SC-West chip first. Then he automatically detects the peaks in each lane. Scout can help with molecular weight sizing by tracking migration distance of your proteins of interest within each electrophoretic separation. And then he can also give you area under the curve calculations to quantify target abundance for each target in each single cell. Scout also offers numerous data visualization tools to display your data. The single-cell Western workflow is fast and easy to do, so you can get results within a day. First, you load your cells into the SC-West chip, then you put the chip into Milo. Milo lyses the cells in the array, does the SDS page separations, and UV immobilization step. And all of that takes about 5 minutes to run. Then you take your chip out and probe it on the benchtop with the antibody probing chamber provided with Milo. Then read your chip out on any open format microarray scanner. Most people already have access to one of these, and there's a list of compatible scanners on our website. We can also package Milo with 2 scanner options that are capable of scanning 2 or 3 color experiments. But no matter what scanner you use, the output of the microarray scanner is a series of images of the chip. Scout reads these images in, does the automated data analysis, and helps you visualize your data. The full workflow can be done in a single day and typically takes about 4 to 6 hours to run. Milo can measure 2 different types of heterogeneity. First, he can quantitate the heterogeneity in the target expression in each of the cells in your sample. So for example, he can tell if there are some cells that have low amounts of protein and some that have high. And this is measured by looking at the variation in the peak area of your target of interest across the cells in your sample. And then Milo can also enumerate to quantify heterogeneity in the number of cells present that are target positive for a particular target. So this is useful if you, for example, if you want to look at and identify subpopulations of cells in a mixed sample by tracking phenotypic markers. There are lots of different ways to visualize your data. So for example, when quantifying heterogeneity in target expression, you might want to create scatter plots of the peak area across multiple samples or chip replicates, like shown in the top left. You can also create histograms of the peak area to see distributions of expression. And when enumerating, you can also create 2D scatter plots and then set thresholds to determine how many cells express a certain target or target combination. And then these can be translated into bar charts, which again quantify the percentage of cells that are classified as different subtypes based on the measured protein expression levels. The SC-West chips can also be archived. When Milo immobilizes a sample into the gel after electrophoretic separation, this process is very high efficiency and quite stable. As a result, the SC-West chips can be archived for up to 9 months after running your sample on Milo. This allows researchers to introduce time delays into their experiment, or come back later and reprobe their samples to ask new questions using the same sample. Getting the most data out of your samples can be particularly important for researchers working with precious primary samples. Milo can also multiplex on a per-cell basis to detect multiple proteins in each single cell. So here you can see some example data demonstrating a 4-plexed experiment. So here we're showing data from a single cell imaged in 3 different colors. So the same cell that you're seeing was captured in the well at the top, and we're just showing images in different spectral channels of the same lane. So you can multiplex with Milo with 3 primary strategies. First, you can spectrally multiplex. To do this, you can probe your sample with a cocktail of primary antibodies raised in different host species. Then just probe with different spectrally tagged secondary antibodies and image your chip in multiple spectral channels. So in this data, we used a mouse, goat, and rabbit primary antibodies, and then used donkey anti-mouse, donkey anti-goat, and donkey anti-rabbit secondary antibodies labeled with red, green, and blue dyes to detect 3 different proteins in this single cell. The second strategy is to use differences in the molecular weight of your proteins of interest to resolve them in the same spectral channel. So here in the blue channel, you can see we probed with 2 different rabbit primary antibodies and the same fluorescently tagged secondary. So the proteins L858R and beta-tubulin are sufficiently different in molecular weight that we can clearly resolve them in electrophoretic separation. And then thirdly, you can strip and reprobe the chips. So once the sample is covalently bound into the gel, it is robust to numerous stripping and reprobing cycles. Up to 9 cycles have been shown in the literature, with about a 50% loss in sample after the 9th cycle. As Dr. Cadet will detail in her presentation, her group has demonstrated a powerful 11-plex assay, which is one of the highest multiplexing levels demonstrated on the Milo platform to date. And so with that, I'll pause there and introduce Dr. Noémie Cadet from the National Cancer Institute at the National Institutes of Health, and let her present her work using highly multiplex single-cell westerns to explore heterogeneity in protein kinase C signaling and CRISPR knockdown efficiency.
Thank you, Kelly, and thank you for everybody to give me the opportunity to present our work. In the experimental section of this presentation, I would like to talk about 2 projects. The first is detection of signal transduction events downstream of protein kinase C at baseline and after activation. And the second is detection of NOX1 knockdown efficiency in CRISPR-Cas9 manipulated LS513 colon cells. Some of the questions we wanted to address in the first project were: how many signals can be detected on a single chip? What is the stripping efficiency, and how many times can the chips be stripped without significant loss of signal? What are the best loading controls to use? What is the reproducibility of signal detection in different cycles? And are there any novel findings revealed by single-cell Western? In our experiments, we have used a well-characterized robust system, the LNCAP cells treated with PMA or DMSOS control. LNCAP prostate cancer cells are responsive to the activation inhibition state of protein kinase C. Formol myristate acetate or PMA activates multiple proteins by binding to their C1 domains and inducing conformational change and translocation. In LNCAP cells, there are multiple PKCs that are activated after PMA treatment, and those include PKC alpha, delta, epsilon, or PKD1, and out of which PKC delta is expressed at highest level and is the most important for signaling. Multiple changes occur downstream of PKC activation, including phosphorylations, transcriptional changes, new protein synthesis, protein degradation. Some of them are weak, some of them are stronger. On the diagram here, you can see the kinetics of these changes. ERK and MEK phosphorylations are early and transient. Phosphorylation of PKC delta and PKD1 are more sustained. The early induced proteins EGR1 and CFOS can be detected after 40 minutes of PMA treatment. PKC alpha gets downregulated after a few hours, while PKC delta levels don't change. In these experiments, LNCAP cells were treated with maximally effective dose of PMA for 60 minutes in order for us to be able to detect both the phosphorylation events and the new protein synthesis. By this time, phosphorylation of ERK and MEK is too weak to be detected. So now that you have learned or you got some sense about what proteins we detect in single-cell Western, let's get to the data. We could successfully detect 11 targets using the illustrated staining, detection, and stripping cycles. We detected different PKC isoforms, PKC alpha and PKC delta. We detected phosphorylated forms of PKC delta and PKD1. We detected newly synthesized protein CFOS, EGR1. We detected several loading controls as GAPDH, beta-tubulin, vinculin, or total ERK. In our experiments, we detected PKC delta in 2 different cycles, cycle 9 and cycle 12 with the same antibody. And we detected CFOS twice in cycle 1 and cycle 10 using 2 different antibodies. We use antibodies mostly from mouse and rabbit, and use stripping total of 7 times between application of antibodies from the same species. A default dilution for an unknown antibody is 1 to 10, but some of the high affinity good antibodies can be diluted 120, 180, as seen here for PKC delta. On the next slide, you can see two lane plots that were generated by Scout that show the separation of all the proteins that were loaded on the PMA chip. On the x-axis, you can see the lane or the well number, while on the y-axis, you can see the peak center for each detected signal. Larger the traveling distance, the smaller the protein is. So, for example, you can see that tubulin or ERG, that are lower molecular weight proteins, are on the top, and vinculin with a molecular weight of about 120 kilodaltons, or PKD1, run much closer to the loading of the cell. These small tables on the right indicate how many cells on the chip contained the signal. This chip was loaded with about 1,400 cells. Let's now see how the signal looks on the chip through a few examples. Here on the left, you can see the CFOS, the inverted image of the CFOS signal from about 1/8 of the chip. And on the right, you can see how the signal really looks, basically having a darker background and lighter spots. You can also see that CFOS was detected as two bands, a higher molecular and a lower molecular weight band, and some cells contain the high molecular weight form, others the low molecular weight forms, or others both. CFOS is known to run as a double band on traditional Western with an approximate molecular weight of 50 to 58 kilodaltons. CFOS can be detected in our samples only after PMA activation, but not in control. Based on how well-known proteins ran on single-cell Western, for example, vinculin or ERK, we calculated that the approximate molecular weight of the higher molecular weight CFOS band is about 130 kilodaltons, while the molecular weight of the lower band is about 60 kilodalton, suggesting that the two bands detected by single-cell Western are CFOS that runs as monomer and dimer. CFOS is a member of the AP1 transcription factor family, known to dimerize with c-Jun and other members of the family. EGR1, the other newly synthesized protein, runs as a single protein on both single-cell Western here on the top or on traditional Western blot shown in the bottom. The third signal that was detected on these chips without any stripping was GAPDH. On the right, you can see a very strong signal for GAPDH that was detected at about 100 kilodalton, which is 4 times higher than what is expected. The molecular weight of the GAPDH is about 36 kilodaltons. This behavior of GAPDH was described earlier in a single-cell Western paper from Kang et al., stating that the exceedingly low mobility of GAPDH in the 4-degree Celsius conditions may be attributable to non-fully dissociated and non-fully denatured native tetramer forms of GAPDH. Here I would like to mention that the cell lysis and running conditions are different for a traditional Western and a single-cell Western, because in single-cell Western, the cell lysis, it's in running buffer that does not contain any reducing agents. It's done for a very short period of time, namely 10 seconds at room temperature. So it's very possible that some of protein complexes might not be fully dissociated and some of the buried epitopes might not be fully exposed. This slide shows for the first time quantitative data from our experiments. It is a modified 1D plot showing all the signals that were detected in the control or PMA-loaded chips. It's a complicated slide, so let me just lead you through it. On the x-axis, you can see all the detected signals, namely ERK, GAPDH, PKC alpha, delta, tubulin, just to name a few, that based on their behaviors are grouped into non-responders or responders. On the y-axis, you can see the level of the signal, namely the peak area, on a logarithmic scale. 0 values are artificially jittered around 0 to be able to plot on a logarithmic scale. The control sample is shown in red, and the PMA treated sample is shown in blue. The green lines represent the mean values, while the height of the diamonds on the right represent confidence intervals. Overall, the data from single-cell Western correlates well with the data from bulk samples detected by traditional Western blot. We can see similar expression of non-respondent signals in control and PMA-treated sample. You can see that on the left for the non-responders, the red and the blue signals are very similar to each other. We can clearly observe target upregulation for modulated targets in PMA sample. You can see higher level of expression for CFOS, phospho-PKD1, phosphoserine 299, or EGR1. In a small subpopulation of control cells, we detect some expression of modulated targets, but at lower level than in the PMA-treated sample. This data was further analyzed by Josh Morho from ProteinSimple, the DataViz. And the PCA analysis identified the subpopulation of control cells that were partially stimulated, as shown by increased phosphorylation of PKC delta and PKD1. In the middle part of the slide, you can see the basal level cells shown in red and the partially activated cells shown in purple. About 30% of the cells, about 100 cells, were partially activated. No such difference was seen in stimulated cells. Cells seem to all be in a similar state after stimulation, which is not a surprise because these cells were treated with fully effective dose of PMA. When the PCA analysis of both control and PMA simulated samples was combined, it became obvious that the partially activated cells shown in purple are more similar to control cells shown in red than to PMA simulated cells shown in blue. If the two samples hadn't been run on separate chips, we might not have been able to identify this small population of slightly simulated cells, as they overlap with the control cells in the combined analysis. Further experiments are needed to evaluate the biological importance of this finding. The next slide shows two other visualization tools provided by Scout to present quantitative data. And right now we are going back to the CFOS signal, where the CFOS was detected in two forms. On the x-axis, you can see cells that are expressing only the high molecular weight form of CFOS. On the y-axis, you can see cells expressing the low molecular weight CFOS, while on the right-hand side, top right-hand side, you can see cells expressing both. The enumeration table on the right shows both the number of the cells and the percentage of the cells that express both or one or the other forms. About 40% of the cells express both the high molecular and the low molecular weight form, while about 12% and 40% of cells express only the high molecular weight or the low molecular weight form. Here on the left-hand side of the 2D graph, you can see double negatives. And if we recalculate the numbers by removing those double negatives, then we find that about 60% of the cells express both isoforms, and 80 versus 20% of the cells express the low molecular weight or the high molecular weight form. Having the quantitative data, we wanted to do some comparisons between or some correlations before different downstream signaling events after PKC activation. First, we compared two phosphorylation signals. You can see phosphorylation of PKC delta on the x-axis and phosphorylation of PKD1 on the y-axis. And you can see on these 2D plots that the two signals correlate very well. Next, we compared the phosphorylation of PKC delta with the synthesis of a new protein, namely EGR1. Again, the phospho-PKC delta is presented on the x-axis, while the EGR1 is expressed on the y-axis. And again, you can see that the two signals nicely correlate. In the next step, we correlated the different loading controls. In all four graphs, you can see ERK presented on the x-axis and GAPDH or vinculin presented on the y-axis, both for control and PMA samples. If you look at the graphs, you can see that, again, the ERK correlates very well with GAPDH and vinculin, both in control and the PMA sample. We don't see so nice correlation between ERK, again presented on the x-axis, and tubulin on the y-axis, especially for PMA-treated sample, most probably because tubulin is affected by PMA treatment. Next, we compared one of the strongest non-responder signal detected with the same antibody in two different staining cycles. These graphs show PKC delta staining in cycle 9 on the x-axis and PKC delta staining in cycle 12 on the y-axis, and you can see how tightly and nicely the two signals correlate. The final comparison as part of the assay validation was to compare the signal, the CFOS signal detected with two antibodies in two different signals. We compared the high molecular weight and the low molecular weight CFOS signals separately. Staining with mouse antibody in cycle 1 is on the x-axis. Staining with rabbit antibody in cycle 10 is on the y-axis. And again, you can see the correlation between the two signals. With every methodology that includes cyclic staining and signal removing stripping, one might be concerned about the stripping efficiency and if any of the remaining signal interferes with or affects the detection of the next signal. In all our experiments, we have used harsh stripping conditions. That meant that we were incubating the chips for 90 minutes at 65 degrees Celsius in a harsh buffer that contained 2% SDS and markup to ethanol. We had very good experience both with stripping efficiency and for not having significant effect on the proteins remaining on the chip. In our hands, it seems that the stripping efficiency is somewhat signal dependent. You can see from the images that we could almost fully remove CFOS and EGR1 with stripping, but we could detect some of the remaining GAPDH signal, the bottom image. Stripping efficiency can be quantitated by SCOUT. On the left-hand side, 1D plots, you can see the level of each signal when we compared the before stripping and after stripping plots. The arrows, the red and the teal arrow, shows the change in the mean and the median of the signal. The table shows the output from the SCOUT calculations that is better visualized probably on the bar graphs at the bottom. Our conclusion was that the stripping efficiency in these chips was pretty good. But additionally, we also try to avoid detecting the same molecular weight protein in consecutive cycles as much as possible. Now we switch to the second project, the detection of NOX1 knockdown efficiency in CRISPR-Cas9 manipulated LS513 colon cancer cell line. And let me just start with some introduction. LS513 colon cancer cells have higher NOX1 expression than the other cancer colon cell line used in the laboratory, the HD29 cells. Both cell lines are used as model systems to study the role of NOX1 in carcinogenesis. NOX1 is NADPH oxidase 1, has an important role in the homeostasis of reactive oxygen species, and it's implicated in carcinogenesis. The goal of the laboratory was to create NOX1 knockout in LS513 cells using inducible CRISPR-Cas9. Single-cell Western was used to guide clonal selection of knockout cells in addition to PCR and traditional Western blot testing. First, we used single-cell Western to detect the NOX1 knockdown level in parental, in empty vector cells, and also in pools of cells that contained T2 or T3 guided RNA. We could observe very low very low percent of ERK-positive NOX1 negative cells, suggesting that clonal selection from the pool population is further needed. About 100 clones were selected from both the T2 and T3 clones and tested first with PCR and traditional Western for NOX1 expression. Selected clones were further evaluated with single-cell Western. This slide shows the data from the Cas9 empty vector clone. And I want to mention that NOX1 is known to be not to be an easy protein to work with. It was detected with a custom-made mouse antibody, and you can see from the left-hand side image that NOX1 runs as a smear on single-cell Western. We used ERK as loading control to identify all the occupied wells. The 1D plot on the top right-hand shows the expression level of ERK and NOX1, while the enumeration table at the bottom highlights that the percentage of ERK1 NOX1 negative cells it's low, it's 8.8% in the empty vector control cell line. With this slide, I would like to mention the unusually high percent of ERK negative cells that have NOX1 signal. The enumeration table shows that we could detect about 14% of cells that express NOX1 but not ERK. Currently, we don't know the biological significance of this finding, but it might reflect high phenotypic diversity of cells observed during cell culture. This slide shows the data from the best clone, the T210. You can see the nice dark image on the left with no NOX1 signal. You can see the nice ERK signal on the right. And even if in this case the analysis was skewed toward detection of false positives because we wanted to make sure that we detect all the NOX1 positive cells, we could detect only a few cells that were NOX1 positive, about 3%. And on the 1D plot, you can see that the expression of NOX1 in these few cells is very low. We could also identify two special clones in which there were many cells, and only one of them is shown here, in which many cells show no NOX1 expression, but about 17% of them have relatively high NOX1 expression as shown on the 1D plot. This cell line showed on traditional Western blot showed almost no detectable level of NOx1 or very low NOx1 level. But with single-cell Western, we could identify that this is a mixed cell population. And with further clonal selection, probably it would be very easy to get NOx1 knockdowns. On the last data slide, I would like to present the quantitative data from parental, several empty vector clones, and also some of the selected NOx1 clones. You can see on the y-axis the normalized NOx1 level is shown on a logarithmic scale, and the red line represents the geometric mean. The lower group of cells contain no NOx1, only ERK. The middle group of cells show expression of both NOx1 and ERK, and the top group of the cells have NOx1 expression but no ERK expression. You can see that the best clone, the T210 clone, stands out from the rest by having almost no NOx1 expression in any of the cells. And from the graph, you can see the distribution of the different cells in these clones, but we don't have an idea about the percentage. The percent of NOx1 knockdown cells is shown in the bar graphs here on the bottom. Almost all T210 cells were NOx1 negative. Also, the T330 cells were almost all NOx1 negative. This data correlates well with reactive oxygen species measurement that is a functional readout for NOx1 in these cells. Finally, I would like to summarize in this slide the main findings from the two single-cell Western projects. We could successfully detect more than 10 targets, including phosphoproteins and newly synthesized transcription factors, using multiple cycles of staining and stripping in a well-characterized cellular system. We identified two states of CFOS in LANCAP cells, possibly representing monomers and dimers, and heterogeneity in cells expressing the low molecular weight, the high molecular weight, or both forms. We identified two subsets of LANCAP control cells, cells at baseline and partially activated cells. We used single-cell Western to guide selection of NOx1 knockout clones after inducible CRISPR-Cas9 manipulation to complement PCR and traditional Western blot screening. Finally, I would like to acknowledge all the people who contributed to these projects, starting with my 20-year-long boss, Peter M. Blomberg, who is now happily retired. Special thanks to members of our laboratory, my great colleague and head of the core, Jessie Chan, our talented summer student, Maria Novitskaya, and our wonderful volunteer, Ramasri Satanore. We worked with Agnes Juhas from the Doroshov lab on the NOx1 project. We really appreciate the discussions and the interaction with Kelly Gardner, Eric Jabart, and Josh Mulho, who are the real experts in single-cell Western and Scout. Thanks for all the support we get from Stefan Swager, Berendia Jackson, and Ann Cron. And thank you for your attention.
Thank you, Dr. Cadet and Dr. Gardner, for your presentations. A quick reminder for our audience on how to submit questions. Simply type them into the drop-down box located on the far left of your presentation window labeled Ask a Question and click on the Send button. Dr. Cadet and Dr. Gardner will answer as many questions as time permits. The first question is, stripping efficiency and how many times can stripping be done? What is the protocol for stripping and reprobing the chips, and how challenging is it to implement?
The stripping efficiency is generally done in hard stripping buffer that contains 2% SDS mercaptoethanol at 65 degrees. And based on our experience, the efficiency depends on the signal that can be objectively measured and calculated using Scout. Although we are very satisfied with the stripping efficiency from the chips, we use this strategy to start with the most important weak signal when we probe the chips and later check the stronger and reference signals. I was not a big fan of stripping membranes when doing traditional Westerns, but we are pretty pleased with the stripping results on the chips.
Thank you. How easy is Scout software to use?
Scout software is used to quantitate signals for each individual cell on the chip, converting images to numbers to enable quantitative comparison correlations. I have experience with version 2 of the software that is easy to use, especially after learning how it works from the online tutorial and spending some time getting used with its logic. The later versions include more automation in detecting specific signal versus the dirt, and one of these features is the auto exclusion, which is a significant improvement as it is very tedious to manually inspect each signal for each cell for several hundreds of cells. The software provides useful visualization tools for data presentation. And as you could see from the presentation, these are lane plots to see chip loading and separation of proteins, histogram, 1 and 2D plots, enumeration table. Plus, quantitative data can be exported as CSV and FCS files for users who want to use their own method to present findings. Additionally, feedback about the software and requests for improvement are welcomed by the software development team.
Thank you. In the presented studies, ERK is used as a reference protein loading control in multiple occasions. In regular Western blots, generally actin, vinculin, and GAPDH are used as loading controls. Could you explain why?
We used ERK mostly because of convenience. It's a clean signal. It has a very nice noise signal-to-noise ratio. The lab has both mouse and rabbit antibodies. It's a low molecular weight signal that often does not overlap with other signals. Additionally, actin it's not efficiently detected on single-cell Westerns. And as GAPDH runs as a tetramer, as you could see from the presentation, and we didn't like to use it that much. Tubulin was affected by PMA treatment, and vinculin could be another option. Plus, the ERK was probably the cheapest antibody that we have.
Thank you. In the introductory slides, it has been stated that the single-cell Western uses Western antibodies. Could you comment on if all antibodies work, and what about the phosphospecific antibodies?
Based on the company's experience, about 70% of antibodies that give a good signal on traditional Western blot work for single-cell Western. In our experience for the PKC signaling studies, similarly, about 2/3 of the antibodies worked for single-cell Western. We had a few that didn't. So, and if we go into phosphospecific antibodies, for example, the phosphoserine 299 antibody worked, but the phosphotyrosine 311 didn't. One explanation for lack of signal on single-cell Western could be that the proteins are not fully denatured because there is no reducing agent present, no heat. It's a very short lysis time, so some epitopes might stay buried, not accessible to the antibody. But in our experience, for the majority of the signals that we wanted to detect, after trying several antibodies, we could find one that worked.
Thank you. How many cells did you load per chip?
The optimal cell loading of a chip is about 1,000 cells on the 6,400 wells, because this one gets enough cells on the chip to address heterogeneity, but possibly minimizes the number of doublets. For the control chips, in this case, about 500, 600 cells were loaded, while on the PMA chip we loaded about 1,400 cells. The standard protocol from the company for single-cell Western workflow is to load 1 ml of 100,000 cells on a chip. This number can be lower, as low as 10,000 cells per ml, or even lower, because it's just you wait long enough and then you can load more and more cells on the wells. Basically, the cells enter in the well based on gravity, and the loading really depends on the number of the cells and the time for loading the chips. Recently, even though the recommendations from the company is 100,000 cells per ml, recently I prefer to load 50,000 or 40,000 cells per ml, especially for cells that tend to aggregate.
Thank you. It looks like you have excellent repeatability and tight correlations for the same antibody probed on different probing cycles, suggesting minimal sample loss over stripping and reprobing cycles. After your 7th cycle, do you think the sample was still stable enough that you could have gone through additional stripping and reprobing cycles to further increase multiplexing?
Our data showed excellent correlation in PKC data signal between probing cycles 9 and 12 on the same chip. This suggests to us that the sample loss has been low enough over the stripping and reprobing cycles that we are still getting accurate measurements of PKC data heterogeneity after 12 probing cycles.
Thank you. Why did you use so many rounds of stripping and reprobing versus utilizing more channels on your 4-color scanner?
In my case, the time was not that important, and I preferred to see every single signal, especially when we were probing them for the first time as a single signal. This one included additional probing and washing steps to probe, not to combine the antibodies, but to detect them separately. But my preference was, again, especially at the beginning, to use them one by one.
Thank you. It looks like we have time for one more question. How many signals can be detected, and is there a theoretical maximum?
I don't think that there is a theoretical maximum in how many signals can be detected. The number of detectable targets probably depends on the antibody species. For example, if one uses rabbit, mouse, rat, goat antibodies, it also depends on how good the antibodies are. It will depend on the molecular weight of the signals and the stripping efficiency. I can see one problem with several rounds of stripping and reprobings as we get to really high numbers, because the chip gets dirtier with multiple rounds of manipulation. Lint and dust from the environment gets on the chip, or there are, with the chip manipulation, there are multiple scratches. So maybe one factor that limits the number of signals that can be detected is basically the chip handling.
Thank you. I would like to once again thank Dr. Cadet and Dr. Gardner for their presentations. I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today will be addressed by the speakers via the contact information you provided at the time of registration. We would like to thank our sponsor, ProteinSimple, for underwriting today's educational webcast. Until next time, goodbye.